In this session, we are going to deal with biomolecules of life. The cells and tissues of virtually all organisms are made up of the same basic molecules. Many of these are substances which are familiar to us, such as carbohydrates, lipids, proteins, and the nucleic acids. Carbohydrates are one of the four major classes of organic compounds in living cells. They are produced during photosynthesis and are the main source of energy for plants and animals. Carbohydrates can be simple sugars or monosaccharides, double sugars or disaccharides composed of a few sugars such as oligosaccharides, or composed of many sugars, polysaccharides. They have a general formula that is CH2O. Monosaccharides are nothing else like it's a white crystalline solid that contain a single aldehyde or ketone functional group. So they are divided into two classes based on whether they are aldehydes or ketones, aldoses or ketosis. They can either also be classified as like triose, pentose, hexose, heptose on the basis of the number of carbon atoms present in them. So aldoses are sugars which possess one aldehyde group and several OH groups, hydroxyl groups. So the most common aldohexose is glucose. Ketoses have one carbonyl group and one carbonyl or a keto group and several OH groups. The most common ketose we use is fructose, means fruit sugar, main mix of sugar. So disaccharides, so two monosaccharide units join together by a glycosidic linkage to form a disaccharide. So some common disaccharides are maltose, sucrose and lactose. In maltose, two molecules of glucose are combined to form maltose. So maltose is the disaccharide produced when amylase breaks down to starch. So maltose is usually found in germinating seeds such as barley. Sucrose is another organic compound. It is commonly known as the table sugar and sometimes also called saccharose. So the glucose and fructose units are joined together. I mean both glucose monosaccharide and the fructose monosaccharide are joined by acetyl oxygen bridge in the alpha orientation to form the sucrose. Lactose, it is also a disaccharide sugar that is found mostly in milk and forms one unit of and it is formed from one unit of galactose and one unit of glucose. So these are the three simple sugars: the monosaccharides, disaccharides, and monosaccharides and disaccharides. Two types of simple sugars. Polysaccharides. So polysaccharides, from the name itself, it signifies that composed of hundreds or thousands of monosaccharides combined together to form these polysaccharides. And these monosaccharides are joined through dehydration synthesis. Some examples of polysaccharides include starch, cellulose, and glycogen. So these are more familiar names to us. They have several functions, including the structural support and storage. So starch is in starch itself we can find two types like amylose, which is a linear unbranched chain of several hundred glucose units formed of several hundred glucose residue units. And the glucose residues inside this, the glucose residues are linked by a glycosidic bond between the first and fourth carbon atom. Same way, amylopectin, it also differs from amylose in being highly branched. Like apart from being 10 residue unit, it approximately the 30th residue along the chain. A short side chain is attached by a glycosidic bond to the sixth carbon atom. So it is amylopectin. So starch can be of two types, amylose or amylopectin. Glycogen. Glycogen is usually found in animals. So it's a multi-branch polysaccharide that serves as a form of energy storage in animals. In humans also glycogen is present, but they are primarily stored in the cells of the liver and muscles. And it can be only used as a function as a secondary long-term energy storage. Thirdly, that is cellulose. It is also an organic compound. It's a polysaccharide consisting of a linear chain of several hundred to over 10,000 beta-linked D-glucose units. So cellulose is the structural component of the primary cell wall of green plants and many forms of algae. So these three are the three main classes of polysaccharides which come across in the day-to-day life. Next section we are going to deal with lipids. Lipids are a broad group of naturally occurring molecules which include fats, waxes, sterols, fat-soluble vitamins such as vitamin A, D, E, and K, monoglycerides, diglycerides, phospholipids, and others. The main biological function of lipids is that they include energy storage, and they can act as structural components of cell membranes and also as important signaling molecules. The lipids may be divided into, commonly they are divided into 8 categories, that is fatty acids, glycerolipids, glycerolipids, glycerophospholipids, sphingolipids, saccharolipids, and polyketides, and stereolipids and phenolipids, both of these. So what are fatty acids? So fatty acid is a carboxylic acid with a long aliphatic tail or chain, which is either saturated or unsaturated. So most naturally occurring fatty acids have a chain of an even number of, mostly they have a even number of carbon atoms, that is ranging from 4 to 28. So fatty acids are usually derived from triglycerides or phospholipids. When they are not attached to other molecules, they are known as free fatty acids. Fatty acids are important sources of fuel because when metabolized, they yield large quantities of ATP, that is adenosine triphosphate. They yield more energy, more quantity of energy can be produced. So that's why they are used in, they can be called as a source of fuel. Fatty acids can be of 3 types, namely unsaturated, saturated, and essential fatty acids. Unsaturated fatty acids are, they have one or more double bonds between carbon atoms. Between the two carbon atoms, there will be more than one bonds. That is, a pair of carbon atoms connected by double bonds can be saturated by adding a hydrogen atom to them, converting the double bond to single bond. So that can be done. Therefore, the double bonds are called unsaturated. The two carbon atoms in the chain that are bound next to either side of the double bond can occur either in a cis or trans configuration. That is, in cis configuration means that the adjacent hydrogen atoms are on the same side of the double bond, whereas in the trans configuration, the bond is the next two hydrogen atoms are bonded to opposite sides of the double bond. So cis and trans is in the opposite configuration. Saturated fatty acids are long chain carboxylic acids that usually have between 12 and 24 carbon atoms and have no double bonds. Unlike unsaturated fatty acids, saturated fatty acids does not have a double bond. Thus, saturated fatty acids are saturated with hydrogen. In an example, we can say that it's palmitic acid or stearic acid can be quoted as an example for saturated fatty acids. Essential fatty acids that are required by the human body, but that cannot be made in sufficient quantity from other substrates or substances, and therefore must be obtained the food that we consume, are called usually essential fatty acids. So two such essential fatty acids are linoleic acid and alpha linoleic acid. They are usually found in the plant oils. Next is category of lipids is glycerolipids. So glycerolipids are composed mainly of mono, di, and tri-substituted glycerols. The most well-known being the fatty acid triesters of glycerol called triglycerides. The additional subclasses of glycerolipids are represented by glycosyl glycerols, which are characterized by the presence of one or more sugar residues attached to the glycerol via glycosidic linkage. Next form of lipids is glycerophospholipids. Glycerophospholipids, also referred to as phospholipids, they are ubiquitous in nature and are key components of the lipid bilayer of cells, as well as being involved in the metabolism and cell signaling, mainly in the cell signaling. The heads of the phospholipids are polar and said to be water-loving, that is hydrophilic. The tails, whereas the phospholipids are non-polar, that is they are water-hating, or we can say hydrophobic. So the glycerophospholipids found in biological membranes are phosphatidylcholine, ethanolamine, etc. And also one more is there, it can be phosphatidylserine. Those three are the commonly seen glycerophospholipids in biological membranes. Next type of lipids is sphingolipids. Sphingolipids are actually a complex family of compounds that share a common structural feature, a sphingoid base backbone that is synthesized from the amino acid serine and a long chain fatty acid called such as acetyl coenzyme. then converted into ceramides, phosphosphingolipids, glycospingolipids, and other compounds. So the major spingoid base of mammals is commonly referred to as spingosid. Stereolipids. Stereolipids such as cholesterol and its derivatives are an important component of membrane lipids along with the glycerolipids and sphingomyelins. That's the same with phenolipids and saccharolipids. Also describes saccharolipids actually describes compounds in which fatty acids are linked directly to a sugar backbone. Next up, biomolecule we are going to deal with is protein, a very important biochemical compound it is. And proteins are biochemical compounds consisting of one or more polypeptides, typically folded into a globular or fibrous form, facilitating in order to facilitate a biological function. So a polypeptide is a single linear polymer chain of amino acids. They are bonded together by peptide bond between the carboxyl group and the amino group of adjacent amino acid residues. That's why proteins are made up of, we say that they are made up of amino acids. So general, the genetic code specifies around 20 standard amino acids in the protein. We'll just discuss about what are the amino acids and what type of amino acids are present in the body. Amino acids are the building blocks for peptides and proteins and play an important role in metabolism. So 20 different amino acids are found in living organisms. They can connect to each other via peptide bonds to form long chains. Proteins may consist of thousands of amino acids and can have molecular weights around up to several million Dalton. The general structure, or you can say the chemical structure, chemical formula of an amino acid consists of a tetrahedral carbon atom connected to four groups, that is a basic amino group, an acidic carboxyl group, a hydrogen atom, and a substituent R group. This R group will be varying in each amino acids. This is the R group, or the substituent group is the one which varies or which differentiates each amino acids. Classification of amino acids can be done in different ways, like that can be based on polarity, structure, nutritional requirement, metabolic rate, etc. But generally they are used, the classification is based on polarity. So based on polarity can divide the amino acids in the following ways, like non-polar amino acids that have non-polar amino acids they have equal number of amino and carboxyl groups, and so they are said to be, as they are equal in number, they are neutral. They are said to be neutral. So these amino acids are hydrophobic and have no charge on the R group. The amino acids in this, the non-polar amino acids include uh, in this group, the amino acids coming under these groups are alanine, valine, leucine, isoleucine, phenylalanine, glycine, tryptophan, methionine, and choline. So these are the non-polar amino groups in which the amino and carboxyl groups are neutral. Polar amino acids with no charge. These are the amino acids that do not have any charge on the R group. So these amino acids, they usually participate in the hydrogen bonding of protein structure. The amino acid groups which come under this are the serine, threonine, tyrosine, cysteine, glutamine, asparagine, etc. Polar amino acids that have a positive charge are have more amino groups as compared to carboxyl groups making it. So the amino acids which have positive charge on the R group are usually placed in this polar amino acid category, with positive charge category. They are lysine, arginine, and histidine. So there are polar amino acids with negative charge also. That is the R substituent group will be having the negative charge on. Those acids are aspartic and glutamic acid. Structure of proteins can be just randomly called. They are not just randomly called chains of the amino acids. They can a variety of intramolecular interactions enables the amino acid chain to fold in a specific way to give the protein a three-dimensional structure and space. A shape. This structure is critical for its activity and function. These structures are contribute for their activities and functions. To describe the complex structure of proteins, we can say that they can be divided into four levels of organizations, like primary, secondary, tertiary, and quaternary. Primary structure, the sequence of amino acid determines the primary structure of the proteins. Like changing at least a single amino acid sequence in a critical position of the protein can significantly alter its activity and function, and a single change in the amino acid can lead to cause of diseases and disorders. So the amino acids connected to each other plays an important role in the structure distinction of the protein. Secondary structure are regular elements. They are such as alpha helix and beta pleated structures we can find usually. Tertiary structure, it describes the complete the three-dimensional structure of the whole peptide chain. Mm, and quaternary structure consists two or more separate polypeptide chains linked together. So other non-amino acid components such as minerals, lipids, carbohydrates can also be a part of protein. So the quaternary structure describes how these different chains and components interact and connect to each other by hydrogen bonding or electrostatic attraction on sulfide bridges. So such quaternary structure example can be quoted as hemoglobin is a very good example for quaternary structure of protein. Nucleic acids. Nucleic acids are biological molecules that are essential forms of life on this planet. They include DNA and RNA. They carry the genetic information of it. Nucleic acids were discovered by Friedrich Mueller in 1869, and the term nucleic acid is the overall name for the DNA and RNA. So nucleic acids were named for their initial discovery within nucleus and for the presence of phosphate groups. All living cells and organelles contain both DNA and RNA, while viruses contain either DNA or RNA, but usually not both. So DNA is called as the code of life. It can be called as the hereditary molecule of the cellular life forms, as it is used to store the genetic information and store and transmit the genetic information. See, nucleic acids, the basic components of nucleic acids include like they are linear polymers of nucleotides. So each nucleotide consists of three components, that is a purine or a pyrimidine nucleobase. Sometimes it's called nitrogenous base also, a pentose sugar and a phosphate group. The substructure consisting of a nucleobase plus sugar is termed a nucleoside. That is sugar plus nucleobase plus sugar. Sugar in the sense it might be a deoxyribose sugar or a ribose sugar which is used in RNA or DNA. It can be a nucleoside. So nucleic acid type differs in the structure of the sugar in their nucleotides. Like DNA contains deoxyribose sugar while RNA contains the ribose sugar. Also the nucleobases found in the two nucleic acid types are different. So, in adenine, cytosine, and guanine are found in both. These adenine, cytosine, and guanine are the purines. They are called nitrogenous bases, and they consist of six-membered or five-membered nitrogen-containing groups. So, purines are six-membered and five-membered nitrogen-containing rings. Pyrimidines, in contrast to that, is a six-membered nitrogen-containing ring. So, these, according to the base components, are in DNA. You can find adenine, cytosine, and guanine are found in both RNA and DNA, while thymine occurs in DNA and uracil occurs in RNA. So the sugars and phosphates in nucleic acids are connected to each other in an alternating chain or through phosphodiester linkages. So the basic purines which are used in the components of in performing the components of DNA and RNA: adenine, guanine, hyposine, xanthine. So adenine and guanine found in DNA and RNA, but usually hypoxanthine and xanthine are not incorporated into the nucleic acids as they are being synthesized out. So the four pyrimidines are uracil, thymine, cytosine, and urtic acid. Nucleotides, you can say adding one or more phosphates to the sugar or a nucleoside will give you a nucleotide. So these are the basic components of nucleic acid structures, which we have to keep in mind while studying the structure of DNA or RNA.